seminar series. We have actually, I think we've got.
Did we just lose him? Okay, we'll give him a second.
No worries.
Come back in. That's funny. Otherwise I can try and quickly introduce you. I don't know if he had something written up, but oh, here he goes. He's back. I got him.
The person that introduced me just disappeared. That's a first.
Yeah. I know, me too. I'm like, what just happened? All right, let's see. He'll probably connect or not. He might be gone again. Okay, hang on.
No worries.
Interesting. All right. I'll go ahead and introduce you. Let's
Okay.
see if he comes back in. I'll let him take back over. I think he's trying to rejoin. So, hi everyone. We just had a little tech issue. Ken's back, connecting to audio probably. Maybe I'll let him
Okay. Can you guys hear me now?
Yep, now you're good.
All right, I got kicked out of here twice. So I will welcome Chris Funk here. I think he's got a really interesting talk today. He's going to talk to us. It sounds like about two projects, right? Like one on a bird species, one on an amphibian. I've known Chris for quite a while now, over 20 years or so, I guess. I knew of his work before I actually met you. I think I met you when you were postdocing at UT Austin with Canatella. I guess you were there for a while. Is that right?
Probably, yeah.
Chris was kind of one of the major players in the early development of the field of landscape genetics, I would say. And so his work, he was working on frogs in the Pacific Northwest. So that really caught me attention from an organismal perspective. But he's since gone on to do, really expand his research in many directions. He's an author of a recently released textbook, right? Conservation Genomics. That's, you partnered with, at least Fred Allendorf was on that as well with you, right? The shameless plug for the for the Conservation Genomics book, which is a really exciting book. It's nice to have a more updated conservation genetics text than the Allendorf one was great, but that was kind of like the late 90s or so. So it's nice to have something more modern around, which Chris played a big role in. So I guess without further ado, I'll go ahead and turn things over to Chris here, and he's going to talk about adaptive divergence and climate change. From a genomic perspective, why evolutionary perspective matters for predicting sensitivity to increasing temperatures.
Thanks a lot, Ken. Thanks for a nice introduction and for the invitation here today. Might have been hard getting there given that we got a little bit hammered here in Colorado with a snowstorm recently, and sounds like you guys are getting some weather as well. All right. So today I just want to start off with a little introduction about what I do and my background and how I got here. I think it's really, I always really enjoy hearing a little bit about people's personal story, how they ended up doing what they're doing and what motivates them. And then I'll give a general introduction to my research topic today, which is the importance of understanding interest-specific trait variation for predicting vulnerability to climate change. Then I'm going to highlight a couple of ongoing research projects in my lab, collaborative projects with a bunch of other folks as well. And the first focuses on the genomics of thermal adaptation in Channel Island song sparrows. And then I'll briefly highlight some emerging results coming out of our research on predicting vulnerability to climate change in the amazing tailed frog. And I'll make a plug for why it's such an amazing species. And then kind of wrap it all up. All right, so first off, I'd like to, let me go back here. I want to talk a little bit about my background and research program. So I feel really lucky to have grown up in the beautiful and ecologically diverse state of Oregon, which I think played a really big role in inspiring my love of nature, scientific discovery, and conservation. So if you haven't been to Oregon before, you've probably just been told that it's really rainy, but actually a good chunk of Eastern Oregon is high desert. So it's really ecologically diverse place. I grew up on a hill here in the Willamette Valley called Cooper Mountain. That's just outside of Portland, about half hour. So it's kind of on the rural urban boundary. And probably like a lot of you and other people that have decided to go into biology, I've always loved being outside as long as I can remember. So as a kid, I spent as much time as possible wandering the forests and streams of Cooper Mountain. And then when I became a young adult, I continued to explore the outdoors any way I could, including backpacking, rock climbing. I was a wildfire fighter for a little while. But my favorite outdoor activity was whitewater kayaking, as rivers were always my favorite environments. So I always felt most at home outdoors, and biology was a really natural fit for me. But then something happened that turned a budding young biologist into a budding young conservation biologist, which was the clear cutting of the childhood forest that I used to play in. I took these photos when I came home from college one summer, and this totally broke my heart and pissed me off. I was a teenager, but it also motivated me to try to make a difference. I've heard a similar story from the late Michael Soule, who's basically the father of modern conservation biology. And he was inspired to conserve wildlands and biodiversity after seeing the natural areas around his childhood home of San Diego destroyed for development. And it turns out that was exactly what was happening to my precious Cooper Mountain. It was getting taken over by the cancer of suburbanization. And although I didn't know it at the time, what I was witnessing was part of a global trend that has been aptly termed the Great Acceleration. So I think these images really capture this unparalleled rate of change in human population size, per capita consumption, all these other things that go with it, like McDonald's restaurants. That's really driving biodiversity loss. And our planet has never endured anything quite like this in its 4.5 billion-year-old history. And one of the most devastating aspects of the Great Acceleration for biodiversity is habitat loss and fragmentation, as exemplified here by slash and burn agriculture. And of course, another aspect of the Great Acceleration that increasingly threatens humans and biodiversity alike is climate change. So we all know that mean temperature has already increased by about 1 degree Celsius since the Industrial Revolution, and is predicted to increase much more by the end of the century, even under the most optimistic scenarios. And unfortunately, it looks like we're currently headed towards one of the more pessimistic scenarios, as exemplified by repeated record global temperatures. So just about every year, you open up the paper, and a little bit after the new year, there's another record temperature that we've made. And this is not the world record we want to break. So this leads me to the goal of my research program, which is to strive to understand the evolutionary ecological mechanisms that generate and maintain biodiversity. And at the same time, I strongly believe it's our responsibility as ecologists, evolutionary biologists, wildlife biologists to communicate biodiversity research with policymakers and natural resource managers so that they can make well-informed decisions. These are people that are trying to make the best decisions possible, but most of the time they have to make decisions without as much information as they'd like. So my favorite projects are those that simultaneously test basic theory and directly inform conservation. My research is really integrative. I combine quantitative field methods, experiments. A lot of my research has a population genomic element, as well as computational approaches to address questions related to biodiversity. And this is why my Twitter handle is Field Genomics, because I still consider myself old school field biologist in many ways. My main responsibility in most of the collaborative projects I'm involved with is to lead the population genomics component. I also work on a huge variety of organisms, as represented by these examples of species I've worked on. And my lab works in a variety of habitats and locations, shown by these red pins. So far, just the Western Hemisphere, from northern temperate forests to the tropics. And as is clear from the Funk Lab logo, my favorite ecosystems are mountains, rivers, and other freshwater habitats. And there are a lot of great things about my job, but I think the best part is mentoring the next generation of biologists. So this slide shows some of the graduate students, postdocs, and undergraduate students who I've had the pleasure of working with over the last several years. And I really try to make a special effort to recruit diverse students and postdocs, because science, this is the pursuit of truth through observation and experimentation and logic, really should be for everyone. And I strongly believe that these diverse perspectives are really essential for tackling the biodiversity crisis. So in terms of research topics, I like to think of my research program as a tree. So the overarching motivation behind the research program is to understand the evolutionary ecological mechanisms generating and maintaining biodiversity, as well as to inform biodiversity conservation, which you can think of as the trunk and roots of the tree. And then there are 4 main branches of investigation in my lab. So one of the main branches focuses on conservation and population genomics. The second branch focuses on climate change, vulnerability, and resilience. A third branch focuses on the microevolutionary mechanisms and environmental drivers causing adaptive divergence. And then a last branch focuses on various aspects of tropical ecology, evolution, and conservation, particularly in tropical South America. And then these can be thought of as specific research projects in the area of conservation genomics. As Ken was just mentioning, Fred Allendorf, myself, Gordon Lukart, Sally Aiken, and Margaret Byrne just finished the 3rd edition of this conservation genetics text, which is now called Conservation and the Genomics of Populations. And we're pretty excited about that. It came out this spring. Another project focuses on testing the mechanisms causing speciation in frogs in megadiverse countries like Ecuador and Colombia. A third project focuses on investigating the relative vulnerability of populations of a very interesting frog called the tailed frog in the Pacific Northwest to increasing temperatures. And then lastly, another one of my favorite projects tests the causes of bind scale or microgeographic adaptation of birds. So obviously I'm not going to talk to you about all of this today, but what I am going to talk about are these 2 projects that have to do with climate change vulnerability. And although these areas, microgeographic adaptation at first may not obviously seem like it's related to climate change, I'll explain how I think it really is very intricately linked with climate change vulnerability and understanding vulnerability. In addition to research and teaching, I also serve as director of a center here at Colorado State University called the Global Biodiversity Center, which has the mission of advancing understanding, conservation, and appreciation of biodiversity. And thanks in part to our efforts, CSU was recently recognized as one of the top 10 universities globally in biodiversity conservation by the Center for World University Rankings. So that was fun. All right, so now that you know a little bit about my way of looking at the world and what motivates me, before I dive into the 2 specific projects, I want to give a little background and theory on vulnerability to climate change and why traits matter for understanding vulnerability to climate change. So, as everyone in this audience likely knows, climate change is predicted to have pretty severe effects on biodiversity. So this figure is taken from a landmark meta-analysis that Mark Urban conducted several years ago. And it shows studies that have predicted the effects of climate change on biodiversity under different emission scenarios. So the x-axis here is different potential increases in temperature over time, and the y-axis is the percent of species in a given community that are predicted to go extinct under these different emission scenarios. So if we're able to meet the policy target of less than 2 degrees Celsius, then the predicted average percent loss is 5%. But we're currently in a trajectory more around here, which will result in an average percent loss of 10% and more. And depending on your perspective, that may not sound like that high of an extinction rate, but keep in mind that this doesn't include the effects of all the other threats that biodiversity faces. So habitat loss, fragmentation, invasive species, disease, pollution, overexploitation, et cetera. But before we move on, one thing to note here is that there's a ton of variation in predictions of vulnerability. Sure, there's a general increased prediction for percent of species expect to go extinct as the temperature goes up, which isn't expected, but there's a ton of variation around this curve. And this is due to real variation among communities and species, as well as a lot of uncertainty in these predictions. So one way we can help move solutions forward as scientists is to do a better job of predicting vulnerability. But what is vulnerability? Let's break that down. So ultimately, one way of defining vulnerability is that it's sensitivity times exposure minus adaptive capacity. So let's define each of these terms. So vulnerability can be thought of as the probability of extinction in response to climate change. So essentially the y-axis in the Mark Urban figure is a measure of vulnerability. Sensitivity is the response of a population or species per unit change in whatever environmental parameter you're looking at. So what's the response of a population per 1 degree increase in temperature? So a lot of times people conflate sensitivity with vulnerability, but they're different because vulnerability is also a function of exposure. So that's the amount of climate change. So for example, the amount of increase in temperature or the amount of increase in variation in precipitation. So ultimately, vulnerability is a product of sensitivity times exposure. And lastly, vulnerability can be moderated by adaptive capacity. And that's the ability of a system to adjust to climate change via dispersal, plasticity, and/or evolution. So if a population has high adaptive capacity due to any of these factors, then it should be less vulnerable than if it has low adaptive capacity. So ultimately, if we want to do a better job of predicting vulnerability, we need to do a better job of understanding sensitivity, exposure, and adaptive capacity. So it's pretty well recognized among ecologists and evolutionary biologists that genomics is a useful tool for at least characterizing dispersal of gene flow, for example, in the field of landscape genetics. That's a big emphasis of that field, and for quantifying evolutionary potential. But it's less well recognized that genomics and evolutionary theory are also key, as I'll argue today, for understanding variation and sensitivity. So focusing for now on sensitivity, to do a better job of understanding how sensitive populations or species are, we need to focus on what I'll call sensitivity traits. So some traits that determine sensitivity to climate change could be things like thermal tolerance is an obvious one, drought or flood tolerance, or if you're a marine organism, particularly one with shells, then a critically important sensitivity trait is tolerance of acidification. And of course, as we already mentioned, climate change is not the only rapid environmental change that populations are experiencing. You can easily imagine traits that determine sensitivity to these other stressors, such as disease resistance. That could be thought of as a sensitivity trait, competitive ability against invasive species, etc. But the take-home message here is that understanding what determines variation and the sensitivity, or conversely, resilience traits, is key for predicting vulnerability to climate change and other rapid environmental change. So going one level deeper, we can ask, what determines variation and sensitivity traits, both among and within species? So my colleagues and I came up with a very simplified conceptual framework to help us think about what's driving variation and sensitivity traits. And this framework starts off with the landscape and environmental heterogeneity that organisms and populations experience during their evolutionary history. And this in turn affects microevolutionary processes like selection and local adaptation, genetic drift, and gene flow. And these processes mold the amount and spatial distribution of genetic variation. And this genetic variation in combination with direct environmental effects determines the distribution of phenotypic variation. And here by phenotypic variation, we're talking about sensitivity traits. And finally, variation in these sensitivity traits in combination with exposure determined by environmental change ultimately determines spatial patterns of vulnerability across the landscape. So the general idea here is that environmental heterogeneity is going to mold the distribution of sensitivity traits via evolution and plasticity. Now, there's nothing particularly groundbreaking about this framework. This is essentially the way that most evolutionary biologists think about the world. But what is different is the focus specifically on sensitivity traits and the variation in sensitivity traits across the landscape and what that means for spatial patterns of vulnerability. All right, so today I want to make the case with a couple of case studies that understanding local adaptation and sensitivity traits is key for understanding which populations are predicted to be most vulnerable, which is essential information for conservation management in the face of rapid climate change, because managers conserve populations or species at the population level. So this first panel shows a very common paradigm in climate change vulnerability assessments in which there's no intraspecific variation in sensitivity traits. So the solid blue line right here is showing current temperature across elevation, but this could be any temperature gradient. And then the dashed blue line right here shows the critical thermal maximum, also known as CT max, which is the temperature at which motor function ceases of a given species across elevation. And then the solid orange line shows expected increase in temperature across elevation under climate change. So if we assume there's no variation in thermal tolerance, as indicated here with the flat dashed line, then these low elevation populations are going to be the first to experience temperatures that exceed their thermal tolerance. So they're going to be most vulnerable. However, if there's local adaptation in CT max, such that the low elevation populations have a higher CT max than the high elevation populations, then vulnerability may be pretty similar across elevation. Or if CT max evolves more at low elevation, as shown here with the dashed orange line, because of greater genetic variation, this is a pattern that's seen in a lot of species and could result in greater evolutionary potential at low elevation, then the low elevation populations may be less vulnerable than the high elevation populations. So you can make all sorts of different hypotheses and predictions, but the important point here is that understanding if there's intraspecific variation in these sensitivity traits and what the pattern is of intraspecific variation is really important for understanding which of these populations are going to be most vulnerable and most resilient to increasing temperatures. So I would argue that two of the most important questions to answer for predicting vulnerability to climate change are: one, how common is intraspecific variation in sensitivity traits? And can we predict patterns of variation in these sensitivity traits? And two, at what spatial scale can local adaptation and sensitivity traits occur? So if there's microgeographic adaptation in sensitivity traits, which is essentially a fancy way of saying that there's fine scale local adaptation within a species dispersal distance, then there could be lots of spatial variation and climate change vulnerability. All right, so that's sort of the framework that I'm working with. So now let's look at some data. Let's look at a couple examples. So first I want to highlight a study that's being led by my former PhD student, Maybelline Gamboa, who defended a PhD a couple years ago and is currently a visiting assistant professor at Colorado College in Colorado Springs. Maybelline was co-advised by Cameron Gambor and myself at CSU, and also by Scott Sillett at the Smithsonian Migratory Bird Center. And another key collaborator is Scott Morrison from the Nature Conservancy. So interestingly, in this case, the impetus for research on the Channel Island song sparrow was a conservation problem, which then led to some interesting basic questions. And oftentimes you start with basic questions in our field and then they have conservation implications, but in this case, it was essentially the opposite. So the Channel Island song sparrow, Melospiza melodia gramineum, is a subspecies endemic to the California Channel Islands, which are right off the coast of Southern California. So LA if you are look at this map, LA is this giant blob, basically where the sparrow is shown. And originally, the Channel Islands song sparrow was found on 6 of the 8 California Channel Islands, but now it's only found in these 4 northern islands. So it went extinct on Santa Barbara Islands, a very small island, in the 1960s, and on San Clemente Island in 1973. Fortunately, the National Park Service, which owns and manages Santa Barbara Island, wants to reintroduce song sparrows to Santa Barbara Island. So before the ranching era, which was in the 1800s through much of the 1900s, the Channel Islands song sparrow bred on Santa Barbara Island. But with the introduction of cats and rabbits, and then catastrophic fires, the final sucker punch, song sparrows went extinct in the 1960s. But since then, the cats and rabbits have been removed from the island, and they've also done a really good job revegetating the island. So it's leaving an island vacant of introduced mammals and also still vacant of Channel Islands song sparrows. They have not recolonized it. So the island is ripe for the reintroduction of song sparrows. But one important question is, which island or islands is or are the best source for reintroduction of song sparrows to Santa Barbara Island? So there's a lot of different options that include San Miguel Island, Santa Rosa Island, Santa Cruz Island, or even song sparrows from the mainland, although those are considered a different subspecies. So there's lots of options, and it's not necessarily a clear decision which population should be the source. So importantly, one factor that should be on the table in terms of making a decision here is a really pronounced climate gradient across these islands. So San Miguel Island here to the west is really directly exposed to the cold California current that comes from the north. It's relatively cold, foggy, and windy. And then on the opposite side here in Santa Cruz Island, it's fairly shielded from the California current. And so it's really hot. It's less foggy and windy. And so going from San Miguel to Santa Cruz, we like to say is like going from Scotland to Baja California. So we also know, based on work by the late Russ Greenberg and also Ray Danner, who's at East Carolina University, the bill size of opportunistically collected museum specimens varies across these islands. So in particular, residual bill surface area, so it's bill area corrected for body size, is greater on islands with higher maximum temperature. And there's also some variation that has to do with the size of these islands. The smaller islands are influenced more by the marine environment. So it's hypothesized that the larger bill sizes on hotter islands is an adaptation for losing dry heat. So bills are highly vascularized, as shown in this micro CT scan, which allows heat exchange without losing water. Also, the bills are warmer than the core body temperature, as shown here in this thermal image, which suggests that the heat is lost through the bill. So Russ Greenberg's results suggested, but do not conclusively demonstrate, adaptive divergence among the islands driven by climatic variation. So our overarching question here is, are song sparrows on different islands adapted to different climatic conditions? So if so, then we might want to use populations adapted to a similar climate as Santa Barbara Island as the source for reintroduction. So we broke this big question into 4 more specific sub-questions. First, do contemporary island populations also differ in thermoregulatory traits? Here we're talking about bill morphology. Secondly, how much gene flow occurs between islands, and what is the relative importance of genetic drift? We looked at this using neutral SNP loci, so single nucleotide polymorphisms. Third, is there evidence for divergent selection among the islands? Got this using what are called FST outlier and genotype environment association tests. And then lastly, are there loci that are associated with bill variation, which we got it using genome-wide association tests? So in essence, we're testing for multiple lines of evidence for adaptation to make as strong an inference as possible, by linking phenotypic variation to the environmental variation, testing whether there are any loci that are associated with the phenotypic variation. And then lastly, testing for divergent selection, which is about, is a pretty increasingly common approach to understand local adaptation in non-model species. So for song sparrows, this means that we're testing for relationship between bill size and temperature, between the temperature variation and specific genes, and also between variation and the bill morphology and specific loci. All right, so to get at this first question about whether contemporary island populations differ in thermoregulatory traits, Maybelline sampled extant populations of song sparrows on Miguel, Rosa, and Cruz. Maybelline was super hardcore in the field and managed to sample several hundred song sparrows on these very rugged islands. And for example, Miguel has very few roads, so she was hiking that whole thing. And so for each bird, she measured multiple bill dimensions as well as tarsus length. So a prediction based on Russ Greenberg's previous work is that bill size should be positively correlated with temperature. And since Miguel is the coldest island, song sparrows should have small bills. And since Cruz is the hottest island, song sparrows should have large bills. So what did Maybelline find? She found that exactly as predicted, residual bill surface area was positively correlated with temperature. And despite all the other factors that could influence bill size, the R-squared value was pretty high at 0.27. So this is exciting, but by itself doesn't demonstrate local adaptation, as this could be driven entirely by phenotypic plasticity rather than constitute genetically based local adaptation. So an alternative hypothesis for the variation in bill size is divergent selection on foraging ability caused by variation in diet, as seen, for example, in the famous case of Darwin's finches on the Galapagos Islands. So Maybelline also tested this hypothesis using a combination of measurements of bite force, seed extraction times, and food resources among islands. And she found no evidence for variation in bite force or seed extraction time on the different islands as a function of bill size. And she also found that the food resources largely overlapped among the islands. So unlike Darwin's finches, the variation in bill size in Channel Island song sparrows is not obviously driven by variation in diet. So our second sub-question was how much gene flow occurs between islands and what's the relative importance of genetic drift? Since local adaptation is predicted to be more likely when populations are large and isolated, this is a really important question. So to get this question, Maybelline generated about 22,000 high quality neutral SNPs. So these are SNPs that do not show a signature of local adaptation or selection, using a commonly used technique known as RAD-seq. And then she characterized population structure using a few different methods, as well as inferring effective population size. She had really nice sample sizes from all the islands, except for this little one out here called Anacapa, which has very few song sparrows on it, and in fact appears to have been colonized in the last decade or so, or perhaps it went extinct and then was recolonized. And then she also had some samples from the mainland, ending up with a total sample size of 453 birds. So Maybelline found high FST values. So these are measures of genetic differentiation between the islands and the mainland. So this suggests that the Channel Island song sparrow is indeed a valid subspecies distinct from the mainland. In contrast, the FST values among the islands are quite a bit lower, but still indicate moderate levels of genetic differentiation for a bird. You know, if you were studying desmog salamanders, like Ken does, you would see higher FST values between samples from different logs 2 meters apart. But these are pretty significant FST values for birds. And also based on the clustering algorithm that's implemented in this program called Structure, Maybelline found strong support for 4 genetic clusters grouped by island, with Anacapa grouped with Cruz. She also found some evidence for limited current gene flow between the islands. So for example, these red lines right here indicate dispersers from Santa Cruz Island to San Miguel Island. So the bird that was sampled in San Miguel, but was assigned back to Santa Cruz Island. And she also found the same thing going on in Santa Rosa Island. So there's some ongoing gene flow, but there's still a fair amount of differentiation. Effective population size estimated using the LD method was fairly large for all the populations, ranging from about 500 on Santa Cruz Island to 1,300 on Rosa. So this suggests that there should be little genetic drift on these islands, and that selection should therefore be effective at causing changes in allele frequencies. So in summary, where we're at right now, the birds on the hotter islands have larger bills, and the populations have very low gene flow and large effective population sizes, which are the perfect conditions for environmental differences to result in local adaptation. So next, Maybelline tested for genomic evidence of divergent selection and loci associated with bill variation, starting with testing for evidence of divergent selection among the islands. So to address this, Maybelline used two approaches. One are called FST outlier tests. So like I said before, FST is a measure of genetic differentiation or genetic differences among populations. The y-axis here is just looking across the genome at different loci. And what we're looking for are those loci that show really high differentiation compared to the background levels. That is a signature of divergent selection. And the second approach to use is called genotype environment association tests, which identify loci that are associated, in which they have an association between the allele frequencies at those loci and different environmental factors when you're controlling for population structure. So what did Maybelline find here? First off, she found multiple loci with signatures of divergent selection among the islands. There were 41 loci that were high FST outliers based on 3 different tests. And there were 34 loci that were associated with variation in temperature based on the genotype environment associations. And 4 of these loci identified as under selection by the FST outlier tests were also identified as being associated with temperature in the GEA analyses. And importantly, some of these outliers are in or near genes related to bill morphology. There's a lot of research that's been done on this before. So for example, some loci were found in genes or closely linked to genes responsible for CAM binding, which has been identified in driving bill morphology in Darwin's finches. Also, other loci were in genes responsible for craniofacial development in humans and mice. So even though climate, not diet, appears to be driving variation in bill size in Channel Island song sparrows, some of the same genes underlying bill variation in Darwin's finches are likely involved in causing bill variation in Channel Island song sparrows. Oopsies. So the last sub-question here was, are there loci associated with bill morphology? And this is really important to determine to what degree this trait is genetically based versus plastic. And what she found is that there are many loci that were significantly associated with bill morphology. This is a Manhattan plot that just shows the evidence for an association of a given locus with bill morphology across the genome on different chromosomes. And interestingly, they weren't all localized in a single or small handful of genes, but were spread throughout the genome. So this is a highly polygenic trait, which is not surprising. Most quantitative traits are. And it also, this analysis explained a really high proportion of the variance in bill morphology, so about 69%. So the variation in bill size is not just due to plasticity. And once again, these genes were in some of the same pathways as those identified using the FST outlier and GEA tests, but are different specific genes. So it provides evidence for multiple genes of relatively small effect interacting that may drive this phenotypic variation. All right, so when you put all these results back into that framework, all the lines of evidence suggest that yes, song sparrows on different islands appear to be adapted to the local climate of each island. And getting back to our original conservation goal regarding choosing the best source population for Santa Barbara Island reintroduction, Maybelline's results suggest that it's important to consider adaptation and choose the birds that are adapted to the current or maybe the future climate of Santa Barbara Island. So for a bird that can easily fly these distances, this represents a case of microgeographic adaptation and a decline in sensitivity to increasing temperatures over incredibly small distances. So these birds on San Miguel that have these small bills that don't allow them to radiate heat very well could potentially be more vulnerable to increasing temperatures than the birds on Santa Cruz Island with the much larger bills. And so also the next time you see a bird beak, just think to yourself, that's not only a mouth, it's also a radiator. All right, so next I just want to briefly highlight some emerging results from a project that's very special to my heart on predicting vulnerability to climate change and the amazing tailed frog. And this project is funded by the NSF Rules of Life program and has the overarching goal of predicting vulnerability to climate change by linking environmental heterogeneity to genetic and phenotypic variation. So one of the best parts of the project is that it's been an excellent excuse to pull together some of my favorite scientists, including Brenna Forrester, my former postdoc on our genomics team. She's now a biologist with the Fish and Wildlife Service, and she's actually now working as the lead conservation genomics expert for the Fish and Wildlife Service. On our physiology team, we had our amazing PhD student, Amanda Cecchino, who's finishing up in a month. Alicia Shaw, my former PhD student that I co-advise with Cameron Gallenbohr. On our stream ecology team, we have Jason Dunham, who worked with the USGS in Corvallis, Oregon, Leroy Poff here at CSU, and then our brilliant simulation modeler, Aaron Languth, who's at the University of Montana. So the overarching objective of this really integrated project is, once again, to identify spatial patterns of vulnerability to climate change and these tailed frogs within and between species using this really integrative approach. So this is that framework I showed you at the beginning of the talk, and we're collecting all sorts of different types of data to get at these different levels in this causal pathway that I'll be talking about today. So our study species is the incredible tailed frog, which is the species I proposed to work on for my NSF graduate research fellowship, but ended up not working on because my advisor steered me in a different direction. But I'm finally getting back to it a couple of decades later. So this frog family consists of just two species. There's the coastal tailed frog here from BC to Northern California, and then the Rocky Mountain tailed frog that's in the interior Northwest and Western Montana and Northern Idaho. It's amazing for a lot of different reasons, including that it is sister to all other frog families and diverged from them over 200 million years ago, which is why I call it the coelacanth of the frog world. The males have a prominent tail, which is used for internal fertilization in fast-flowing streams, hence the name tailed frog. And the tadpoles have a sucker mouth that they use to attach themselves to rocks in these streams, where they're amongst the most important grazers. So really important in terms of stream ecosystem function as well. But the main reason we are focusing on them is that they're adapted to cold mountain streams and are thought to be particularly sensitive to increasing temperatures. So the sub-question I want to focus on now is this part of this conceptual framework, and it's whether there's evidence of local adaptation and sensitivity traits and or genomic data in tailed frogs. So right now I'm focusing on this part, and we're specifically testing whether there's any evidence of thermal adaptation across elevation or stream temperatures. So to address this question, we started by focusing on one low and one high elevation site along each of two elevational transects in both species, so 4 sites per species. Here, I'm just showing the study design and results for the Rocky Mountain tailed frog in Montana, but we use the same design in the coastal tailed frog in the Oregon Cascades. And what we've found so far is that summer stream temperatures vary a ton, with about a 10 degrees Celsius difference in the warmest summer temperatures between the warmest and coldest streams. So importantly, this really high level variation sets the stage for potential adaptation to the local thermal regime. So next, Amanda tested whether CT max likewise varies among streams as expected based on this temperature variation. And what she found is that tadpole CT max differed significantly among the streams and varied exactly as expected based on the observed variation in summer stream temperatures. So that's cool. And now we're in the process of testing for genomic evidence of thermal adaptation across elevation. So far, sequencing of 40 individuals from the 4 Montana sites has yielded 34 million SNPs, many of which are related to CT max and or temperature variation. So this means that CT max is genetically based and highly polygenic, and that there's evidence of divergent selection among the streams with different temperatures. So this provides genomic evidence for thermal adaptation. And here's another Manhattan plot showing evidence for association of a given locus with CT max across the genome. And although we're still in the early stages of looking at which specific genes and modifiers are related to CT max and temperature, some of the obvious ones already popping out, such as heat shock protein modifiers, I should say, that are shown on this Manhattan plot. So both the CT max and genomic results indicate that there is indeed evidence for thermal adaptation to stream temperatures, which means we can reject the hypothesis that thermal tolerance does not vary as a function of elevation. So we found some results for the coastal tailed frog in Oregon as well. It's a little bit different there. But importantly, this means that this traditional view that sensitivity traits don't vary within species is not correct in tailed frogs either. So we're excited about these results, but still have a lot more work to do, including genotyping way more individuals across our study regions in Montana and Oregon. So in particular, we will genotype another 272 tadpoles from another 22 sites. So we'll have 15 sites per species across the temperature gradient and the elevational gradient. And we're doing that using a targeted capture approach known as MyBaits. And ultimately, we're going to tie these field physiology and genomic results together using simulation modeling to identify spatial patterns of climate change vulnerability. And in the end, we'll end up with some kind of map that looks a little bit like this, where it shows relative vulnerability across the landscape. And ultimately, the goal here is to understand local adaptation and the sensitivity traits and what it all means for understanding spatial patterns of vulnerability to climate change. So this is what we say we're going to do on our NSF Rules of Life proposal that funded this. But sometimes things happen that are not in your proposal, such as catastrophic wildfires. And once in a millennium heat waves, of course, neither of which are once in a millennium anymore, but instead are occurring more and more frequently, exactly as predicted by climate scientists. And both of these events landed directly on top of our study sites. So in particular, 2 of our sites, shown here by the 2 red triangles in this big pink polygon, were engulfed by the 173,000 acre Holiday Farm fire, while the rest were only affected by the heat wave the next year. So tragically, multiple towns along this highway were completely destroyed by this fire. And this green triangle up here, which is the H.J. Andrews Experimental Forest, LTER site, where we base a lot of our research, narrowly escaped being torched. It just touched the western corner. So through pure determination and stubbornness, Amanda was able to slog through this massive inhospitable burn and find our temperature loggers, as well as tailed frog tadpoles for which she measured CT max. So in the heat wave only sites, Amanda found that summer temperatures shown by the red points were a little bit higher immediately during and after the heat wave. So the blue is before and the red is after. But in the fire plus heat wave sites, Amanda found temperatures were much higher than after the fire than before. And in fact, about as high as we have seen in any of our streams. Similarly, Amanda found that CT max increased significantly in the heat wave sites, but CT max increased dramatically in the fire plus heat wave sites shown here. So the question is, what mechanisms are causing this increase in thermal tolerance? Is this just due to acclimation, to warmer temperatures, in other words, phenotypic plasticity, or is it due to rapid genetically based evolution of thermal tolerance due to mortality of individuals unable to tolerate those temperatures and unable to survive in the much warmer streams? So we were actually funded by the NSF RAPID program that's specifically designed to take advantage of these sorts of catastrophes and situations to address this question. And we're once again going to use a very integrative approach that involves field, experimental, genomic, and modeling approaches. And I also just want to emphasize once again that these extreme events are no longer rare. So for example, as part of a different RAPID grant that was funded about 5 years ago to test the community and genomic effects of an extreme 500-year flood event on stream insects in the Colorado Front Range, where many sites experienced their normal annual average rainfall in less than a week. So it's more important than ever to have long-term monitoring programs in place to test organismal responses to the increasingly frequent and intense extreme events at the genetic, population, community, and ecosystem levels. So what can we conclude from our research on Channel Island song sparrows and tailed frogs? So first off, at least in these two cases we looked at, interspecific variation in sensitivity traits is quite pronounced, suggesting it could be common in other groups as well. It was also somewhat predictable based on environmental variation and what we'd expect based on evolutionary ecology theory. And moreover, the sensitivity trait variation we're seeing is occurring at very small or microgeographic scales. So this suggests that there's a lot of variation in climate change vulnerability as well. So I would argue that we have to stop thinking of species as fixed entities with fixed traits. Local adaptation is powerful and can result in pronounced intraspecific variation in sensitivity traits, which has super important implications for which parts of a species range will likely be impacted by climate change and other environmental stressors first. All right, so that's all I want to say in terms of my research. I just want to finish my talk with a little message of hope and empowerment, especially for the younger generation of scientists in the audience who are inheriting these unprecedented challenges of, we had a global pandemic, we got climate change, looming on the horizon, biodiversity collapse. And I think this point in history is best summarized by J.R.R. Tolkien when he wrote, "I wish it need not have happened in my time," said Frodo. "So do I," said Gandalf. "And so do all who live to see such times, but that is not for them to decide. All we have to decide is what to do with the time that is given us." So for whatever reason, you happen to live in the most critical 20-year period in the 4.5 billion year history of our planet to make a difference for life on Earth. It sounds melodramatic, but it's absolutely true. So it's essential that you as a scientist are at the table where decisions are made. More and more scientists are stepping up to fulfill this responsibility or making a huge difference. This is just a handful of some of my colleagues who are engaging in policy and working with conservation managers who I'm really proud to call my friends. And getting involved and engaged is difficult, but it's also really necessary and also can be really rewarding. So just one little example. So I was invited to a small meeting on adaptive potential at Durham University in England a few years ago with lots of great evolutionary geneticists, but our group was probably the only one that specifically laid out a plan regarding how to make genomics useful in conservation policy. This work is tough, just learning to speak the same language as the policymakers. So we had 2 people from the Fish and Wildlife Service on this paper, and you take some time to understand the language each team is using. But the papers attracted a lot of interest, and now we've been asked by the Fish and Wildlife Service to lead workshops on integrating adaptive potential into the U.S. Endangered Species Act. So don't forget, society needs scientists more than ever right now, and you can make a difference. So thanks again for the opportunity to talk with you about our research, and I hope to meet, well, I guess I won't meet with you later today, but I'm happy to take any questions. And also, if you have any questions by email, I'd be happy to take those as well.
Okay, great. Thanks for a great talk, Chris. We'll open it for questions now. Obviously, certainly feel free to unmute yourself if you'd like to talk, or Chris, you could open the. Sometimes people just prefer to plunk something in the chat window here. It looks like there's
Okay.
one there already.
Cool.
If you'd like to go from there.
Let's see what we got here. How warm is Santa Barbara compared to the other islands? How long did it take to do all the genetic and morphology work in the Channel Island finches? So first question, how warm is Santa Barbara compared to the others? In general, it's an interesting question because Santa Barbara is a very small island. It's quite a bit smaller than the northern islands. And that means that its climate is influenced a lot by the surrounding marine environment. So it's probably somewhere in the middle. It's not quite as cold and windy as San Miguel Island, but it's definitely cooler and probably very variable compared to Santa Cruz Island. So yeah, I mean, there's definitely potential source populations from the extant populations to use to colonize Santa Barbara Island. And one idea is to take individuals from multiple source locations and introduce them on Santa Barbara Island and let selection figure out who's best adapted to the climatic conditions there. In terms of how long it took from start to finish, it took Maybelline probably five years, something like that. So it was a fair amount of work, and she was a very hard worker.
I mean, this is probably, I'm pretty sure I know the answer, but you've read a lot more about this than I have. I'm wondering if you could, comment a little bit on, so you have these S traits, right? And so there's variation there. Of course, a big question, from quantitative genetic standpoint is like, so how much, heritability is there, and, what the time might be for a response to selection. I don't know if you have any sense of that. Probably not for like a lot of these traits, but at least, maybe ones and more similar traits and more model organisms, so to speak.
Yeah, no, that's a great question. So the GWAS study, the genome-wide association studies, is a way to kind of get the question of the genetic basis of a trait in a non-model species. And it's not exactly estimating heritability, but it is indicating that there are a lot of genes that are underlying these traits, so suggests that they are highly polygenic. And so there's actually been some theoretical work recently, I think motivated by this question of evolutionary rescue and whether populations can evolve to rapid environmental change and if it's going to help them persist. Showing that traits that have a simple genetic basis with just one or a few loci that underlie them, they can evolve faster, but they also, the amount they can evolve maxes out faster, versus a trait that's highly polygenic. It evolves a little bit slower, but it can keep on evolving because there's so many gene combinations that can increase the breeding value, the trait value. So that's not directly addressing your question. I mean, I think ultimately we have to see by monitoring these populations. And for example, the tailed frog study with that fire, we'll be able to test whether genes that we've already identified related to temperature adaptation, those alleles increase in frequency rapidly after that fire due to some of the frogs that are the tadpoles that cannot handle those temperatures dying. And so it could just be like a quick selection event. But I get the sense based on theory and the polygenic nature of these traits that they can evolve, perhaps rapidly, but we'll see if it's rapid enough to buffer them from population declines and extinction.
Thanks. There's a couple more there in the chat if you want to.
Yeah, so there's one from Madeline. Is there a reason why we wouldn't want more subspecies to be forming? I'm not sure what the downfall would be if the species altogether can still breed. So I guess I don't totally understand that question. Madeline, do you want to elaborate?
So yeah, so my genetics background is like in the distant past now. So I guess I'm just curious, like, if these subspecies are they problematic at all for genetic diversity within populations? I don't know if, like, if these two subspecies were to overlap, like, is there some genetic downfall for their, survivability or anything that would be a reason that we wouldn't want these subspecies to be forming?
Okay, are you asking about the case of the frog or the bird?
The the bird, the
Okay,
house
so
pigeon.
are you asking like if we were to take individuals from the mainland subspecies and introduce them to the island subspecies, if that could cause some sort of problem?
Yes.
Okay, yeah. So yeah, I think what you're talking about is outbreeding depression, where if you take individuals that are too distantly related and introduce them to a population, it could result in them being maladapted or having some genetic incompatibilities. And that can happen in some cases. More and more research is suggesting that outbreeding depression is usually unlikely, unless the populations are really divergent, or unless they have like a chromosomal inversion that can result in hybrid incompatibilities. But in most cases, when they take individuals from a source population and introduce them to a target population, for example, if it's declining, they want to boost genetic diversity in a strategy known as genetic rescue, usually that's really effective for increasing genetic diversity, reducing inbreeding depression, increasing evolutionary potential, and also population persistence. But it is, when you're going to make those sorts of decisions, you have to weigh the risk of outbreeding depression versus the risk of not doing anything and allowing inbreeding depression to continue to drive down fitness. Does that answer your question, Madeline? Hopefully.
It does. Yeah. Yeah, it does.
Okay, cool.
Thank you.
So there's a question from Jaron. I'm really interested in the idea of sensitivity traits in the context of plants. Do you know what plant traits are or could be important climate sensitivity traits? Yeah, plants have lots of traits that are really important for determining sensitivity to climate change. I'm not an expert in plants, but for example, I have a colleague here, John McKay, who's a plant evolutionary geneticist, and he studies the genetic basis of drought adaptation in plants, which is clearly a hugely important trait for plants. Given that plants they can't hide from selection. They can't find micro habitats that they can walk to and go hang out in the shade or in a wetter place. So they're directly exposed to harsh environmental conditions, and there's lots of adaptive variation in plants. I think drought adaptation is the main one that comes to mind, but I'm sure there's traits also related to temperature tolerance and I guess variation and precipitation is ultimately sort of a drought trait. But you should look up John McKay's work on adaptation to drought. Sam, half-baked question: when it comes to prioritizing conservation efforts, is there any trade-off between spending time predicting a population's vulnerability to climate change and measuring their actual response to it? So doing kind of what we've done with the tailed frogs and putting a lot of effort to try to predict vulnerability versus just going out there and seeing what happens, I think is what you're getting at. You said, obviously, these aren't mutually exclusive, but what are your thoughts on the balance? Seems like the latter might become more possible important as climate impacts accelerate. Yeah, I think our tailed frog study is an example of how these are not mutually exclusive, because we started out looking at spatial variation and these sensitivity traits and trying to predict what that means for vulnerability long-term. But then in the process of doing that, we had a real world, probably affected by climate change, catastrophic event land right on top of our populations, which is now allowing us to essentially test whether there is evolution in response to that event and what that means in terms of persistence of that population. So I'm an advocate for having the studies in the field and expecting that there's oftentimes going to be some extreme events nowadays. Like, I think it'd be a cool project to map the probability of different types of extreme events across some landscape. And I think there's going to be very few places in the world that are not going to be impacted by events that used to be once in a millennium. And Claire, thank you for the talk. What type of genetic samples were collected for the birds? I'm trying to assess gene flow between piping plover populations using buccal samples. So I'm curious if you had any luck with non-invasive samples. So we actually use blood, but there's a lab here at CSU that's run by Kristen Ruig. She has a project called the Bird Genoscape Project. And a lot of their work is using non-invasive samples, and particularly they use feathers for a lot of their work. So what they'll do is sort of a hybrid approach where they'll have really high quality DNA for a subsample like blood and get really good genomic data for that. And then for the remaining samples, they'll just use the base of the feather, and it's not as good DNA, it's not as high quantity DNA, but they're getting enough data from that to do low coverage, whole genome sequencing. And since they can align that to the high quality genomic data they have, that hybrid approach works really well. So you might want to get in touch with Kristen Ruig and talk to her about strategies.
Okay, well, I guess, yeah, if there are any more questions, we'll go ahead and thank Chris again here for giving us a really enlightening and sort of forward-thinking talk. Appreciate you, coming out and spending some time with us. So thanks for a great talk, Chris.
Yeah, thanks a lot. Really appreciate the opportunity. Thanks for your great questions, everybody. Have a good weekend.